I suggest we start with the non-clinical discussion. Do you agree with us?
Microphones are on.
Yes, for the non-clinical, in general, we agree with the comments made by the speakers. Surely it can make sense, as pointed out, for the companies to seek for central advice if there are differences detected between the biosimilar and the reference product on the non-clinical level, to be sure that the development is going in the right direction. I'm not quite sure that we can put this in the guideline from a legal point of view. Perhaps we may ask for advice from the EMA on this.
I believe we can always, I'm not EMA, but I think we can always put a recommendation to the guideline. And the question is whether it's necessary really to put it everywhere, and we should consider this. And again, I believe that companies do have experience with this, and as was highlighted, actually the major issue with the in vivo studies might be the ethics committees, that they want to see in vivo animal studies rather than the regulators. I think that's really obviously a major issue also in the EU, major remaining issue. That's what I heard from different participants today.
So are there any further comments on the non-clinical part of the guideline?
Just on the one question Dr. Weiss just mentioned, and it was only our experience, and coming in from the originator field into biosimilars, I actually suggested doing more preclinical work to try to aid the review at EMA and the progression into clinical trials. And in reality, that did not help with the regulators because, again, the preclinical is not as sensitive to your point. What I was amazed at, though, that same information which was not helpful to EMA was, in fact, really key for us in terms of going into the clinic. And that is an issue that we really do have to deal with from an ethics committee education and communication. So with that statement, I'm not saying that the guidance needs to be different. I'm just saying the realities that what we experienced in the field, that's all.
Thank you. It's Narayan from Biologicals Advice. Is it possible that animal model is a better one for looking at the differences in immunogenicity between two proteins? Because it's no issue of concomitant immunosuppression and so on, comorbidities and so on and so forth that can influence the immunogenicity development. What does the experts think about it?
Yes, my name is Ben Dijkmans. I'm from EULAR, but speaking for myself. First of all, the story of the extrapolation is a kind of a dream. And I agree that first you must work on the mechanism of action, but you have to realize that in rheumatology, our best products, methotrexate, we still don't know the mechanism of action. The second is that almost all our drugs, including anti-TNF-alpha, comes not from rheumatoid arthritis, but the idea was that it was sepsis. So thinking about a mechanism of action, it is somewhat exaggerated in my opinion. The second is the PK, the pharmacokinetics. In rheumatology, it is very difficult to look for a strict correlation between, say, the serum level of an anti-TNF product and the efficacy. So I think also that is somewhat exaggerated. And for all other drugs, MTX, azathioprine, there is no correlation. And the third is that I think you need always clinical studies. And these clinical studies are based in rheumatology, in rheumatoid arthritis, on the OMERACT criteria, and it is the 20, but it is overall the ASA 70 and the ASOS in ankylosing spondylitis. So I think you need still always clinical studies.
I would like to challenge this view, and I can do it because we are old friends with Professor Dijkmans.
That's the reason for my criticism, yes.
Yeah, I share the view that rheumatology is an area where we have many drugs and we don't understand how they work. But I think the situation is different for biologicals. For biologicals, we have a much better understanding because we know the targets much better than for the chemicals. And I think that the TNF inhibition in a broad term is the mechanism of action of TNF inhibitors. And this is something that happens quite late in the inflammatory process, not in the very early phase when the disease mechanisms may be very different. So I would really challenge the need for clinical trials in all indications, but we do require these justifications anyway, and if we have, let's say we have the anti-TNF product, we would, and this is antibody, we would immediately ask which active sites are crucial for the effect of this antibody. And that can be, to a certain extent, investigated in vitro studies. So that's the idea that we don't have to go to the clinical trials in order to extrapolate, except in exceptional cases.
Perhaps this is Mark McCamish again. On that same issue, I think we also have to reflect, and I think it was Dr. Weiss earlier that was pointing out that we've learned a lot in terms of the way that we've dealt with originator molecules. And you know, Novartis is an originator company as well as Biosimilar, and we do manufacturing changes, as do many other companies. As well. And so these are principles that have been developed over time on comparability. So in the rheumatoid arthritis area, for EULAR and then other associations, ACR, these compounds, these biologics have undergone manufacturing changes. Many have undergone several. And some of these have had significant impact on glycosylation, and they went through comparability exercise. And so the science around that is understood and has been used on the regulatory side. So just being transparent in this, and with those changes, appropriate data was required for them to then have that change and receive extrapolation to all indications. So again, it's information that is it's not unique, nor is it new in the field.
Dr. Markus.
Yeah, thanks. I was going to just quickly address the original question that I don't believe animal models actually really inform immunogenicity in humans. Well, most of the animal models are rather contrived anyway, and we have different versions of biologics, humanized or chimeric, et cetera, that would behave potentially differently in animal models than humans. So I don't think I would give too much credence other than, you know, something to look at early, but it won't be definitive. And then just to agree with Dr. McCamish's observations that ethics committees, and it's not just a matter of education of ethics committees as to what the regulatory requirements are and why maybe a trial application is being proposed without data on animal models. also put yourselves in the investigator's shoes, and the investigator needs to feel comfortable with the molecule and comfortable injecting it into their patients. So again, I don't think the guidelines need to change. I think that's just a reality that we all faced in developing biosimilars.
Narayanan? Sorry, thank you. It's Narayan from Biological Advice again. Yeah, I agree with you that immunogenicity in animal model doesn't necessarily translate into immunogenicity in the humans. But my point was, this is a comparability exercise. My point was, can you find if there is a difference between the two products in an animal model, which might become more apparent in animals than in humans, and would actually tell you something to go back and look at the products a bit more carefully. That is the point, really.
So are there any comments on this point, perhaps also from the drafting group?
Yes, yes, this is Gabi Reichmann from Paul-Ehrlich Institute. I think if the molecules were so different that they would elicit a different immunogenic response in animals, we would also be able to pick up these differences in the analytical quality assessment. So I don't think that the animal models, animal data would add anything to the quality characterization.
Yes, Leon van Aerts from the Dutch Medicine Agency. I would like to add to that especially for monoclonals, if we have to do animal studies, we often would go to non-human primates. And one thing we have to realize about such studies, that the number of animals are really limited. And we also see variability in immunogenicity, immunogenic reactions in these animals, not only in monkeys, but also in other species. So you may find some differences between the biosimilar and the reference product, but it's very hard to interpret such differences and if they're really meaningful. So I don't think you should do animal studies for that goal.
Yes, over there, please comment on this.
Yep, Sumant Ramachandra from Hospira. So I think when we do this assessment from a non-clinical as well as clinical level, we have to also be aware that when we are sizing our, whether it's a non-clinical study or a clinical study, for lack of difference, even though you're looking for a difference, the more you look at a variety of models, whether it's non-clinical or clinical endpoints, you're prone to type 1 error. Okay, so you will get this false positive when you're actually looking for lack of difference. And my concern is that unless we're very clear what you're looking for, you're going to just find something, go chase it down a rabbit hole, and don't even know what it really means. And so you have to be very clear as to what we're looking for, both in non-clinical models as well as in the clinical endpoints. I know we'll get to the clinical section, but one of the key questions will have to be on the clinical side. If you're going to go down and do a study, you have to size it to the point that you have to know that in the number of endpoints you're looking at have to ensure that you don't end up in a type 1 error situation.
Any other comments or questions related to the non-clinical part? Well, you can still do those questions, no problem. But now we are open for clinical questions. And while you are thinking about it, I can take one from the Adobe. So this is Pekka Kurki speaking. Hello, everybody in Adobe. The question is quite tricky. What if a licensed biosimilar turns out to be more efficacious afterwards? Does anybody want to play a regulator and give an answer? Personally, I would think that it is highly unlikely, because why would anybody do such a study first? And the next issue is that what could it mean? Is the biosimilar or the reference, were they different in the beginning, or is this something which is due to the evolution? Now, the regulatory view here is that that would be very interesting information, and it would be a kind of difficult thing to explain. But as regulators, we can always ask a new benefit-risk evaluation, but it would be very hard to reject a product if the benefit-risk is improving. So I think there might be some other regulatory aspects, but Dr. Van der Plas has another view.
Thank you, Martijn van der Plas, Dutch agency. I think this is apparently it's an issue that fascinates people, because I think it's a variant which has been on the table 3 or 4 times before today. If a product is more efficacious in a clinical study, and the clinical study is indeed sufficiently powered to say that this difference is reliable, then you should must look back, because apparently there is a quality difference behind this. I mean, differences in efficacy do not come out of thin air. They have a pendant in the quality aspects of the product. And if you find these, then you should go back to the quality characterization sections and see if you have missed something there. And this is possible. Of course, it's always possible. Thank you.
I think that Dr. Markus was waving.
Yeah, my understanding or guess from the question is that the study, if it was an equivalent study, that in essence means you've looked at non-inferiority and non-superiority, and it in essence failed on the non-superiority test and hence was declared superior. So if that's the context of this, then I think it's an interesting challenge. We all say it's unlikely, but that's the reason we do an equivalent study in the first place. It's unlikely, but it's equally unlikely that you'd fail on the inferiority side if your analytics and pharmacology and everything else matches up perfectly well. So I think I think any of us doing biosimilars, if we think we have a high quality biosimilar that matches analytically and functionally and kinetically and every other way, we'll be surprised if we fail on efficacy in the confirmatory study. That's why it's confirmatory, but it's possible on the upper side or the downside. So I think we still have to be open to that. And if it happens, then it's real. We shouldn't pretend it's not real. There's some other feature of the molecule that needs to be fully understood. And I think the reason regulators had put in equivalence instead of non-inferiority is if it has increased activity, then also need to consider increased safety, on target, off target, other aspects of the molecule beyond that of efficacy, which, as you said, enhances the risk-benefit profile, but it actually shows that it's not a similar molecule and might need to consider either an innovator pathway or at least heavily scrutinize the safety data. Thanks.
I think you made an interesting point, because I interpreted the question as such that there would be statistically significant increased efficacy. But you touched upon a different point, that you just are not within the equivalence margin, which is a little bit different. It doesn't automatically mean that a biosimilar would be more efficacious. There's just a higher chance that it might be more efficacious. And while we haven't seen such a scenario, of course that would create discussion, and we would have to look at all data that have been submitted for this product. But if there is a statistically significantly better efficacy, you should identify the reason. It should have been obvious from the very beginning, from your characterization, that there is really something very different. I think it's not easy to show really significant better efficacy, and even with maybe major changes to the molecule. I don't know. But I think that's a different scenario you are touching upon. And we might see something like this in the future. I cannot exclude this.
If you find real that the biosimilar is more efficacious. Is not the practical solution that you say, well, it is more efficacious and we do a large controlled trial instead of looking for 1 or 2 years what must be the explanation? Because you have, in that situation, you have a sign that you have a better product.
Well, the regulatory problem is that it's not possible to switch the status of the regulatory status of the product from biosimilar to a standalone application. And if somebody claims that a biosimilar after licensing is more efficacious, that means that there has to be a randomized clinical trial behind this claim. Otherwise, we would probably not react to it. The question is then, should we do yet another trial to get an answer to this? It might be a hard sell. I would rather ask for a benefit-risk evaluation and maybe ask both the innovator and the biosimilar whether they have changed anything, because the reason may not be in the biosimilar product, but the innovator product. And again, if safety remains acceptable, I think it's a luxury problem.
I think we are discussing here quite extensively a very unlikely scenario. That's my impression. I think it's highly unlikely, but it's obviously very fascinating to talk about this possibility. I think we should, if anything, I would, as a regulator, be more concerned of seeing inferior products, not seeing superior products, not seeing products with really largely increased efficacy. I think this is quite unlikely to happen. But still, we're spending a lot of time on this. I think it's, as I said, I think it's quite unlikely if you have done a good characterization and a really thorough comparability exercise to see really such fundamental differences in efficacy. Then something must have been gone completely wrong in your comparability exercise then. And also I could not imagine such a dramatic change in the efficacy of the reference product, that all of a sudden the reference product has changed, that there was a manufacturing change which induced such a drop in efficacy, and the regulators have not noticed this. I don't know, I think it's a very unlikely scenario. I really cannot imagine it, but who knows? There remains some small risk maybe, but I think we are over. It is a little bit exaggerated, I think, this concern. That's my personal opinion.
I propose that we leave this topic for time being. There is a question in Adobe. It says that in terms of extrapolation to all indications, is it an absolute must to test the most sensitive indication? What is meant by the most sensitive indication? Is there guidance on this? I think this is an excellent question. I think we were eager to say that you have to pick up the most sensitive indication, and then we start thinking, okay, it might be the most sensitive for efficacy, but might not be most sensitive for safety or immunogenicity. And now we have to make some compromises. Anyway, the model that we pick up for confirmatory clinical trial must be sensitive, so that we should be able to detect differences. But it need not to be the most sensitive if it is otherwise the most suitable model. I cannot give any better answer, but maybe you can comment on this. Yes, Dr. Markus.
Thanks, Richard Marcus with Amgen again. So I'd encourage the agency not to be too afraid to ask a sponsor for more clinical data. It's possible, and I had in my presentation, that it's possible that immunogenicity or safety sensitive population is different than that of efficacy. And when going to extrapolate to all other indications of use, I think it's appropriate to get data that's at least sensitive in both, and it may not be the same study. It may be a different design study. One might be larger or smaller than the other, but having that data in a sensitive population to support extrapolation pre-approval, I think, should be required, and the agency shouldn't be afraid to ask a sponsor to go do a second clinical trial. Thank you.
Maybe, this is Dr. Kurki, maybe I can respond to this. We try to stay cool and not react to all kind of scenarios. As regulators, I think we need to be sure that we are not asking for something which is really, it is not really useful. And we end up in this situation many times that we have some uncertainty after the first pivotal trial in aiming at extrapolation. And then the question is, what kind of additional data we might want to have. And often it turns out that you would have to do a tremendously larger study than the initial study to get any meaningful data to support extrapolation. It would be nice to have good pharmacodynamic endpoints, but that's often not, those are not usually available. And if we are not convinced that it would be needed, then we probably wouldn't ask it. Nevertheless, companies often do these studies without us asking for it. But I think as regulators, we should not add to the burden unless we think that it's really justified. More questions? There is one non-clinical question, and Dr. Heim will address that.
Yes, we have received a question whether newer animal models with transplant components of a human immune system could be helpful to build a bridge to human immunogenicity assessment. So I give a word to Gabi Reichmann to give a preliminary comment.
Yes, this is Gabi Reichmann. There are different models, animal models that have human cells or may represent or contain or are tolerant to human proteins, and it may be different. They may be used in a different way or useful in a different way for the immunogenicity assessment. If we are talking about animals that are immunosuppressed and then they are reconstituted with, let's say, human PBMCs, I would say that these animal models are set very artificial, and with regard to the exact components that are reconstituting and the time of duration for that they are reconstituting, that I would not consider them very useful for immunogenicity assessment. There are other models used where animals express a human protein, and in that way they would see this as their own protein, and they would be tolerant to this protein as would be a human that expresses this protein. So in that specific scenario, you could come in with, let's say, if you have mice tolerant or expressing human interferon, you could test the immune response of these transgenic mice against human interferon. And there are actually data available that indicate that different immunogenicity or immunogenic profile of different interferons can be revealed. However, here again, the quality of the drugs that elicited a different immunogenicity in these transgenic mice was so there was a clear quality difference. So if you had done a good quality characterization of the products, you would actually have seen differences, and you would not have needed the immunogenicity data to see that there's a difference in the molecules. So again, these in vivo models, in our experience, would not add additional value.
So thank you for this statement. Are there any comments from the audience on this issue? Yes, Naeem?
Thank you, Narayan, again. I wasn't actually advocating animal study for immunogenicity, actually, in case I misunderstood. It is just a rhetorical question. I think most animal studies with complex biologicals are a waste of time anyway. But in terms of doing a human study that's powered to detect a difference in immunogenicity, I don't think that's practical, to be honest. I think it'll be a nightmare and highly insensitive. So we're left with a situation where you really can't tell with certainty in a pre-approval situation about the difference in immunogenicity unless you do an enormous study.
Hi, Steve Flatman, Lonza Biologics. On the same theme, I wondered whether the EMA had an opinion on the value of in vitro immunogenicity testing models, essentially looking at the activation of T cells and B cells, I guess, whether they thought that data was useful, I guess, particularly in a quality environment.
Maybe we will address this question to our immunology experts. I'm looking at Dr. Thorpe and Dr. Wadhwa. Who is taking the lead?
Yes, so addressing the last question, I think such studies are usually done following in silico type of investigations. And these provide information concerning T-cell epitopes of different types, perhaps. I think they're very interesting scientifically, but they don't really address the issue of whether the molecule is really going to be immunogenic or not in patients or recipients. They might tell you something about what T cells might recognize, but they don't actually tell you whether that process is actually going to happen. And I think the important thing to realize is that those studies are not going to really affect the need to do immunogenicity studies in humans, because they just don't address that at all. So they're not a cop-out for getting you out of doing an immunogenicity study. They may be valuable, although I think you have to be very careful in kind of product identifications, leads. If you've got 10 possible products and some of them have lots and lots of this T-cell activity and others don't, it might be worth considering which ones you develop. But again, I've heard more recently that you can come a cropper with this one as well. So I think basically if you've got a lot of money to spend, do it, but be very careful with how you interpret it. Is that all right? Is that all right to answer? Oops.
Thank you very much. Yes, please.
Michaela Fliego-Kamenczak from Baxter. I would like to add on the immunogenicity. What is your expectation with regards to duration, number of exposure, pre-approval, post-approval, and to add on a subgroup analysis? What would you expect pre-approval?
Thank you. I'm looking at my colleagues who will be volunteering to respond to this. In terms of numbers, we usually say that all patients participating in the clinical trials should be tested, and that usually is a sufficient number for immunogenicity as well. The importance in doing this is that then you can relate those immunological findings to clinical findings. I wonder if my colleague Marie-Christine Bielski has a comment on this. No, she hasn't at this point. Then we are often asked as tell us how you can, what are your requirements for immunogenicity? And then
we will respond that we cannot tell you any numbers because the immunogenicity is not only the number of ADA positive patients, it's the duration of the response, the titers, neutralizing capacity, association with clinical findings, and so on. So it's very difficult to put a number on those. Professor Dijkmans.
Pekka, is there a super statistician in the room who can tell me if you do a comparative trial to look for antibodies against TNF-alpha blockers, how many patients you have more to do a clinical study on efficacy? Because you have perhaps a percentage of 17 against 10 in your power analysis with regard to antibodies. So if you have planned to do a study, a comparative study based on antibodies against TNF-alpha blockers, perhaps that may be enough to do a clinical study also on efficacy.
Dr. Bielski.
MHRA, Margaret Bilski. I think that it's almost clear. I think it's more or less in the guidelines that we are not requiring immunogenicity study powered to show detect a difference. And I agree that maybe we are rather relaxed, but as already said, it's after having checked that all the other data are supportive of biosimilarity, because I agree with you, it's absolutely impossible. It might be possible when the product is very immunogenic, but when you have only 1 or 2% of antibodies, it's impossible. So we are asking for description, we are asking for incidence rate, for titers, and very importantly, we're asking for data on the impact on the efficacy and safety, and usually we are expecting to find the same kind of impact with the two products, reference and biosimilar. And with all this package, we are reasonably confident that the immunogenicity is similar, but there is no way that we expect immunogenicity studies powered to show similarity. And I have to say that initially the FDA text was a bit, we questioned them because we had the impression that they expected studies really to show similar immunogenicity. And during our conversation, I think it was realized that it was not possible and they would not expect that either, but that's my interpretation.
Yes, Dr.
I
Weiser.
think if we see antibodies in the percent range, we usually have a good idea from the clinical trials whether they're similar or not. I think about the growth hormones. We had a very good idea that they were highly similar with regard to antibody development, or definitely for insulin, we should be able. Of course, if they are very rare events, then it's highly unlikely. But still, the biggest concern being in the epoetins so far. Still here we have one example where neutralizing antibodies were caught in a clinical trial. So what we are really looking for is whether we have a problem that we have a product that might really have a big problem, not so much whether it's really the same with regard to immunogenicity, but we want to rule out that there are products come to the market that have a major problem with immunogenicity.
Thank you, please.
I would like to come to Michaela Flego-Kamensek. What's the duration of clinical for the immunogenicity? Which duration of the clinical trial would you expect pre-approval? And to add a second question, what do these immunogenicity data in one indication mean for those indications that are extrapolated?
Does Dr. Giesen want to comment on this duration of the follow-up?
Yeah, with the duration, it's I can't give a firm answer as you already have expected, because it's also dependent on a lot of issues. If it's chronic use, it's if the patient's only receiving it once or twice. So, and it's also normally we say we need with chronic administration we need 1 year immunogenicity data, but we have accepted, and I think that's also reasonable when we don't see any major problems. When we don't see any problems, that we accept 6 months of immunogenicity data pre-approval, and then the other 6 months post-approval.
Yes, Dr. Weise.
I'm sorry, I talk so much. I think the experience gained with the reference product is very important. Also, when the antibody development is expected. For example, with epoetines, we know it takes longer usually to see the antibodies than for an insulin. So we want to look longer for epoetines, and I think the guideline says you should have 1 year immunogenicity data for epoetines. And although we know that there is a huge variability around when they really appear, but with insulins we are pretty confident after 6 months already. So it really it depends also on what you expect from and what you know from the reference product. And we now have a much more tailored approach also with regard to immunogenicity assessment.
There is a question in Adobe. This is Dr. Kurki. This is related to the discussion on extrapolation. And the question is, can we ask a randomized clinical trial post-marketing? And maybe I will forward this to Dr. Giesen first.
Yeah, of course we can ask for it, but I'm not sure if it's very useful, because we have the pre-approval phase 3 clinical trial in which we show similar efficacy and safety. What I would like to advocate is that for biosimilars, that most of the post-approval commitments that are that the reference product has to do, the biosimilar has to do that as well. So if there are registries ongoing for the reference products looking at very rare adverse events, I will strongly advocate that the biosimilar will also be part of that registry so that we have probably collect more cases. But I don't think it's very useful to do post-approval clinical trials if we don't expect any problems.
Maybe the one issue behind this question was also that if we hesitate in the extrapolation, could we then compensate this uncertainty by asking a controlled clinical trial? And I believe that if there is uncertainty which cannot be answered with anything else than a clinical trial, we would probably ask the study pre-licensing. Are you in agreement with this statement? Yeah, colleagues are nodding. Yes, please.
Keith Watson, Parexel Consulting. To your last point, does that suggest that CHMP would not allow approval of a biosimilar with restricted indication?
Well, thank you. That was a clever question. Well, this issue has been discussed. And from the legal point of view, a biosimilar can have fewer therapeutic indications. We always say immediately that this is not desirable for pharmacovigilance point of view. But from the legal point of view, it is possible. At least the legal experts of EMA have confirmed this. Now we are trying to screen what is in the Adobe.
Meanwhile, we have a question from Dr. McDonald.
Hello, Judith MacDonald from Pfizer. I'd like to ask something which is more in the regulatory area. As we've just been talking about preclinical and clinical data, and I think it's a relevant question to consider how much of that preclinical and clinical data on the biosimilar and the reference product actually is then expressed in the label, in the summary of product characteristics, and whether what we do in that regard is actually meeting the needs of physicians, patients, and so on, because we know that from Tajani that the physician and patient confidence is key to sort of the uptake of biosimilars. And what we're seeing now is that the recent policy from the EMA is to label these products as generics, as just including the information on the reference product without the information on that may have been generated head-to-head on the biosimilar, and relying on the EPAR to fill the gap. So really the question is that, have patients and physicians been consulted on that? Do they find that useful to go and look in the EPAR? Are they going to be able to find what they need? And we in EBE did arrive at a position paper this summer, actually recommending, in our view, that the data generated both on the biosimilar and on the reference product should at least be mentioned in the label, because that's much more transparent, and then you can go for further information in the EPAR. So does the EMA plan to consult with stakeholders on this subject?
Well, this is a good question, and we understand the need for biosimilar specific data in the SMPC. But the problem is that what should we then put in the SPC? And I think we are speaking about the section 5.1 pharmacodynamics, which usually contains everything what has been done clinically as well. And the problem is that, as already mentioned by many speakers here, the basis of the biosimilarity is in the physicochemical and in vitro biological characterization and the functional testing. So if we leave that out and only give the clinical data, that would be just part of the story, and that would be immediately misunderstood. And that's why we have the reference to the EPAR, in order to give the full picture to the reader. Well, some busy readers might miss that connection, but if you really want to know what has been done with the biosimilar, then it's at EPAR. And I would really ask everybody to advertise EPARs when you discuss with clinicians or patients, because there you have the whole story.
But has a consultation with the stakeholders happened on, whether they actually find that useful? Because I also think they would actually have to look at the EPAR for the reference product and the biosimilar to work out what studies were done where. And although it's not for the EMA to rule on advice on switching, things like that, physicians have to make those decisions. So perhaps they might find it useful to know that a recent head-to-head study had been conducted, that was at least mentioned in the label, and then they could go for further information to the EPA.
Now I'm looking at my regulatory colleagues. What do you think? What is more useful? And can we describe the biosimilarity exercise in a concise way, as it's expected to be in the SPC? Okay, we have Dr. McCamish and then You over there.
Sure, I guess I would relate it back down to why is the question coming up now? Why not include this information about analytical characterization of the times that you've done manufacturing changes? And why not ask the stakeholders about that then? Why does it come up now? Because manufacturing changes certainly have happened. And it gets back down to there's been a regulatory determination that the pre and post manufacturing product are the same, deserve the same label. And so from a biosimilar perspective, same types of data are there. So to me, again, and then you push back to the stakeholders and the patients. And when you first talk to clinicians, they really don't understand biologics, much less biosimilars. And there's a big educational requirement we've addressed and suggested. That educational requirement is also necessary for patients as well. So this is actually a disservice and would then be a sense of confusion. And I think having the EPAR that they can refer to, including physicians, which is also available for major manufacturing changes, has been appropriate and is appropriate moving forward.
Sumant Ramachandra from Hospira. Actually, to support what Mark just said, the education component is going to be very important because we've actually heard the exact opposite happening in the field where physicians are being visited upon and being pointed out to the EPAR and telling the prescribing physician, look, there's only one study in this indication with the biosimilar drug. It's not being studied in your other indications or your field of interest. Why would you use it? And that really points out to the fact that the point about the biosimilar pathways being missed and the physician is being misled to think that these drugs are not being tested appropriately in the field of interest that the biosimilar companies should test it across all the range of indications. We've heard that from physicians in the field, and I think it's very important, just like the US FDA took the mission several years ago when there was a lot of noise about generics in the field, because all these arguments were still the arguments that people were talking about small molecules 10, 15 years ago, that small molecules cannot become genericized properly. And while it's a very different field, and I agree on the variability and that the biologics are made in living systems, the same level of confusion is being sowed in the field in the mind of the clinician. And those who are clinicians in the room know that those who are prescribing have to take care of patients, and they're not always reading the latest in literature every single day because they're going every 5 to 10 minutes to another patient. So I think it's very important to ensure that the regulators, along with the industry, does the appropriate amount of education, that this field gets traction. Otherwise, it's a disservice that's happening out there in terms of wanting more and more and more data when the prescribing physician is being visited upon.
Professor Laszlo.
Well, also on this issue, we have discussed it some time ago in the CHMP, and I would not say that our discussions have been concluded on this issue. However, the general view currently rather is that the purpose of the label for the physician as well as for the patient is to be informed about the disease and the treatment advised for treating a specific condition. And therefore, it's in our view, more useful to have the information that is also generally available on the studies that have been conducted with the originator, because the biosimilar is indeed intended to replace the originator drug. And along the lines as we have done with the generics, we would currently rather foresee it useful to include data in 5.1 on the study from the originator, and not so much on the comparability exercise, because indeed it may, especially for patients, but also for physicians, it may be very difficult to understand and to swallow this information if it's given in a very abridged way within the place of an SBC.
Thank you very much. We have another question. Yes, please.
As you were just referring, or Josie Dratwell, IPOPI, you were just referring to the EPAR and the fact that healthcare professionals are very busy people and might not have time to read it. Now, actually, the biosimilar word is not mentioned until the second page of the EPAR summary, so it might be an idea to perhaps to prioritize it and put it on the first page.
Yes, indeed. I think there is a question over there. Who has raised a hand there? Oh,
Did you have another question?
yes,
Could I
Judith.
maybe come back on a few of those points that were raised? I think just on the manufacturing information, why, to Dr. McCamish's point about why should that not be in there as well? I think, you know, we have a clear structure from a regulatory point of view of the SMPC in Europe, and we have, you know, there is no section for that in the SMPC. And it seems that normally whatever clinical information is filed with the application is reflected in the SMPC for, it's about the actual product. So in this case, it's about the biosimilar product. But actually there's nothing about the biosimilar product in that SMPC. It's all on the reference product. So, you know, is it very transparent that that is the case when you've actually taken part of it and said, oh, people will misunderstand that, so we'll just put that in the EPAR. And there could be, you know, information on comparative immunogenicity, for example, which the physicians may find useful from that head-to-head study that they would like to see in the label. But I think the point what I'm trying to say is, has it been assumed what patients, physicians, etc. want, or you know, have they actually been asked and consulted?
Well, as you know, every SMPC goes through the testing, but that's maybe another thing. Anybody wants to respond to this? Dr. Weise?
Actually, you say that everything that has been done with the product should be in 5.1. I think that's not true. Anything that is relevant for the indication and for how the indications have been studied and supported by clinical trials, I think that should be in 5.1. And even then, not everything will be in 5.1, only the really important clinical trials. And also, we don't have results from bioequivalence testing in the generic, for the generics in 5.1. So that would be a similar approach. Why don't we have it in 5.1? It's not relevant to the prescriber. This information is not relevant, really. It's what supports the indication. That's relevant to the prescriber.
Another quick point. Do you not think that labeling the products as generic in a generic way gives a sort of counter message? So on the one hand, we're saying they're not generics, and then we are labeling them as though they are. So does that not cause confusion?
I think you misunderstood me. I was talking about real chemically derived generics, and we don't put information on the bioequivalence study in 5.1 for the very same reason, I guess. It's not relevant for the prescriber.
Yeah, no, I understand that.
Yes, please.
Sorry, just to address for Dr. McCamish, and it's actually surprising how in this session, at least, things are pretty well aligned with regards to our views of the guidelines. I think our side, you know, I think we wanted a little more illustration or understanding of the thought process, knowing that specifics may not be available, but when they are, have them available. But my comment more about the last comment of the thought process, and that representing a biosimilar as a process change, I think we all here understand that's not true. It's inaccurate and misleading, and we shouldn't be, I think, discussing or thinking about regulatory decisions or labeling and all of those elements, thinking about a biosimilar manufacturer as a process change. So I'm a little bit concerned about how frequently I hear this and how common it is that it just comes off the tongue and just says, it's just like any other process change. Well, it's phenomenally different from any other process change. So we all here understand that and know that, but when it's discussed that way externally, when you launch, it's your own product. If you were to detail or discuss this with healthcare providers, it shouldn't be discussed as it's just a process change, because it's just not true, and it's deliberately misleading.
So let me respond to that and challenge as well. I don't think I've ever said that manufacturing change is the same. What I've said is the science around it and what can be used and what we've learned from manufacturing changes is applicable to the development of biosimilars. And in fact, in your statement earlier, which is actually a huge false claim, was that when you make manufacturing changes as an originator, you make them to improve the product. You make it to improve the safety, or you make it to improve. That's not the case. Manufacturing changes are done to have a specific impact. Generally, you transfer it from one place to another. You may increase the productivity, you may increase the yield, and in your case at Amgen with Aranesp, you changed the master cell bank to change the process, which resulted in significant requirements for clinical trials and testing. So in terms of the spirit of transparency, I think we have to be clear there as well. The motivation around manufacturing changes is not to improve the safety of the product or efficacy. And what I'm saying, and what is consistent, I think, with the guideline, is that comparability includes tools, science, that is applicable to the development of biosimilars.
I think Narayanan has a question.
No, it's a very quick comment. It's a sad fact that not every doctor reads every SPC in any detail, and that's a reality. So I think education is a key, and educating the key opinion leaders, I think, is the most important, because they're the ones who are going to influence how biosimilar or a similar product is prescribed.
Thank you. There are a few questions in Adobe. There is one question we don't dare to answer, but I will present it to you. The question is, does the EMA think that the lack of firm guidance from the US FDA on biosimilars is holding back the development of biosimilars globally? So as said, we don't take any position on this, but what do you think? Dr. Marcus.
Sure. I'm from the US, so I'm happy to pretend I can address that. But no, I don't think it does. I think sponsors, there's a lot of people here who are manufacturing biosimilars or developing biosimilars with global intent, and any sponsor can still go have a very meaningful discussion with the FDA and get very meaningful feedback without a guideline being finalized in the US. So I don't think it affects development.
That's good news. Dr. Thorpe.
I was going to say the same thing. So,
Okay, there's a question, please.
Just a final statement to this. Actually, what we have seen is that since the law was passed in the US in 2010, the FDA has actually taken a very active path. Their hands were tied before, so it's not like they could have done anything before 2010, before the Affordable Care Act actually passed. But since then, they've actually taken steps, and I've seen an evolution at the FDA, frankly, that has been quite accelerated as they've gotten the staff in place, as they've now started charging fees since last October. So they're actually collecting fees from sponsors as part of the process. So they will catch up. It's just a matter of timing. Europe has been far advanced of the FDA, and hopefully with European and US discussions at the regulatory level, the US will come to some level of hopefully global harmonization. I don't think the key is having good biosimilar discussions with the US FDA, but is that the highly regulated markets and the regulators actually are harmonized in terms of what the expectation on the sponsors are. Because if they're not harmonized, we as sponsors are going to be stuck between a rock and a hard place of trying to develop a biosimilar 3 different ways, one for Asia, one for Europe, one for US. And my own desire is to see the regulators on one playing field in terms of guidance.
Thank you. I think this is good news. And I think we can say that we have good collaboration with the FDA, between FDA and EMA. So I think there is goodwill on both sides to facilitate global development. Professor Laszlo.
Sorry, just along the same lines, I wanted to say that actually we have seen a sort of revival recently of common or joint scientific advice procedures between FDA and EMA, specifically on biosimilar monoclonal antibodies. And while unfortunately that procedure does not end up in a common advice position, it still provides a very early very early discussion and exchange of views, so that chances of ending up in a joint development are, I believe, are significantly increased and therefore valuable.
Thank you.